Dear participants, welcome you all in this course Systematics of Angiosperms. I am Dr. Gausia Bashri, Assistant Professor from the Department of Botany, Aligarh Muslim University, Aligarh. For this course, I am assisted by a teaching assistant, that is my PhD scholar Muhammad Soban Ali. So, first of all, I would like to start this course and welcome all of you in this course Systematics of Angiosperms. Today we will learn about the evidences in plant taxonomy, because as previously sir has discussed about the what are the taxonomy, he has explained the basics objectives of taxonomy. So it is very important to distinguish different families from each other, and for this there is a several evidences which supports this taxonomic relationship of the plant particularly. So today we will discuss the two evidences, that is the morphology and anatomy. So let's start our talk. Now moving forward to the content that we are going to study today, that is the first I will introduce about the topic, then we will study the morphology in relation to taxonomy, what are the evidences of morphology, then I will discuss the anatomical evidences in relation to taxonomy. So moving forward to our first topic, that is the introduction. As we have discussed earlier that the plant taxonomy is a branch of science which deals with the identification, nomenclature and classification of plants. So for this identification we need a description of plants. That is a very important plant. Without description we cannot identify any particular plant, and identification after there is a nomenclature of that particular plant, and then we classify the plant according to their different characteristics and their qualities. So in taxonomic evidences, it is a information used. In this particularly, in the evidences of taxonomy, it is the information used in context for the purpose such as the identification and classification. They will further provide the additional informations to classify or identify a particular plant. So for this, as I have told you, in taxonomy, the evidences is the information which is used to context for the classification and identification of plants. So it is a very important aspect that help to identify or classify a particular plants. So for this evidences, we will discuss this into following categories. Like the first one is the morphology, that deals with the external characters. That is a very most important type, because so the first thing that we have, that is the morphological characteristics. For all the other things we have to further study, and that needed time. But first of all, first thing that we have in the taxonomy, that is the morphology of particular plant. So it deals with the external characters that we can see from some character from our naked eyes. Then the next is the anatomy, as the name indicated, the study of structure of a organism and their part. So this also needed a section cutting. So it is a further steps in the identification and classification of particular plants. Then the palynology is the branch that is study of the pollens and spores. In embryology, we study the embryo, that also help in the work as evidences in the case of a taxonomy. Then next is the cytology, which is the study of structure and function of cells. And the next in this category of evidences is the phytochemistry, which is the study of phytochemicals, which are chemicals derived from the plants. Then the next branch that help in the supporting the plant taxonomy is the ultrastructure, like the architecture of the cells and biomaterial that is visible at higher magnification. And the last and the most important thing is the genome analysis and nucleic acid hybridization, which mainly today we will rely on these technologies, because now if we have to study the phylogenetic studies, this help in the systematic identification of particular plant species. So the identification, measurement and arrangement of genetic features are based on these techniques. So these are some different branches of botany that help in the taxonomic identification of particular plant and to classify them. So now moving forward to the first, that is the morphology in relation to taxonomy. and this is mainly deals with the external characteristics of plant such as habit, root, stem, stem habit, then leaf structure, type of inflorescence, flower type, then perianth structure, their arrangement, type of endoecium, their adhesion, cohesion, then the characteristics of stamens, gynoecium type, carpel structure, types of ovules, fruit types and seed types. These are the some characters, that is the morphological characters that are used to categorize the different plants, and it is a very broad topic. That's why for this particular topic I have created a pura whole 1 week lecture for this particular topic that we will study before starting the families, because if we don't know the morphology, there is a very difficult to define a particular family, their characteristics. So this whole topic will be studied just before we starting the different families, so that will covered in a separate week. So the morphology, that is a very important because it provides the basic language for plant characterization, classification and identification, and this is the most initial step as told you, because from naked eye we can easily see the morphology of the plant. What is the arrangement of leaf, what is the arrangement of flowers, that is called as the inflorescence, the seed type, we all these characters we can see from our naked eye. So this is the most basic language of characterization of the plant, and then this gross morphology provide the foundation of a plant taxonomy, and it is supplemented by the information from various other fields of botany. So first of all, starting from the next, that is the anatomy in relation to taxonomy. The application of anatomical data to phylogenetic problem is a great value in elucidating the taxonomic relationship, because anatomy is further added the information in this particular morphology and phylogenetic studies. So this anatomy is further categorized into the two categories, vegetative anatomy and floral anatomy. Vegetative anatomy deals with the parts of the plant body that serve mainly to the individual's life, like the root, stem and leaf. And the floral anatomy is the essential part of the flower, means for the reproduction, like all the parts of the flower like calyx, corolla, endoecium and gynoecium. So the first one, that is the vegetative anatomy. There are several characteristics that present in the vegetative parts that help into identify the different groups of the plants, that are the epidermis, that then the part of the epidermis, that is the stomata and trichome, these both are the part of the epidermis. Then stem anatomy. In stem anatomy there is a further categorization is the neural anatomy, which also help in the identification of some families. Then leaf anatomy, then sclerates, these are the some specialized tissue, then specialized cell and the cell content, and the wood anatomy. These are the some vegetative anatomy that help in the identification of the particular families or plants. So starting from the epidermis, epidermis is the outer layer of the plant tissue that shape and protect the plants from the damage. It is a protective layer. So it is a shape and wall thickness, wall sculpturing and the inclusion in the epidermal cells. So there is a different types of epidermis depending on the different families, like papillate epidermis cells in case of a Gramineae family, and the presence of distribution of silica bodies in case of a Cyperaceae. Then sclerification of the wall of epidermal cell in some genera, like in case of a composite node is termed as the Asteraceae, and presence of very narrow epidermal cells in case of Styleraceae. Then Sharma and Shyam describe the taxonomic importance of silica bodies in 22 Indian species of cypress. So this is the shape and wall thickness and sculpture, that you can say there is a verification in this particular different types of plants. So this variety of the types of epidermis can help in the identification of particular species or families. As you can see, this is the different types of epidermal cells that present in the plant cells in this particular figure. Now moving towards the next anatomical feature, that is the stomata. Stomata are very useful anatomical tool in the field of plant taxonomy for resolving appropriate position of the several taxa. Stomata is also part of the epidermal cells, as I have already told you, and stomata have been classified according to their origin of various stomatal cells and the position of subsidiary cells and guards in relation to their aperture. So morphology and ontogeny, number and arrangement of subsidiary cells and their relationship with other epidermal cells are characters of taxonomic significance in case of a stomata. As you can see in this particular picture, there are different types of stomatal apparatus found in angiosperm. The first one, that is A, which is called as the anemocytic type, which is in which the epidermal cells around the meta is not differentiated. You can see here all the epidermal cells are similar types and they will not differentiate into other epidermal cells of the layers. Then the second type is the parasitic type. They have a two or more cells parallel to the guard cell. As you can see here in this particular image, the subsidiary cells are just parallel to the guard cells. So these are the termed as the parasitic type of arrangement apparatus. Then the next one, that is the C, that is the diacitic, in which the subsidiary cells are at the right angle to the guard cell, as you can see in this particular image. Then the next one is the anisocytic, that has the three subsidiary cells. As you can see, these are the anisocytic, in which there is a presence of a three subsidiary cells 1, 2 and 3. Then anisocytic type with stomata surrounded by a circle of radiating cells, as you can see in this particular image. They have a circles. You can see there is a number of cells which is surrounding. This is also a anisocytic type. They have a maybe third number or maybe a number of cells. Then this one is the F, which is showing the tetracytic type with 4 subsidiary cells. As the name indicates, they have a 4 numbers of subsidiary cells. As you can see in this, they have a 1, 2, 3 and 4 number of subsidiary cells. And then is the cyclocytic type. As you can see in this one, this is the cyclocytic type which have a concentric rings of subsidiary cells, as you can see in this particular image. Then the last one, that is the graminaceous type. As you can see, they have a dumbbell shaped guard cell with 2 or small subsidiary cells, as you can see here, that have a parallel to the guard cells. So this is the graminaceous type which present in the most of the Poaceae family members. There are some stomatal apparatus type in the which present in the case of angiosperms. Depending upon this stomata, there are the following types of their development as named, that is the ranunculaceous or anemocytic, that is also called as the irregular cell. In this type, feature of the ranunculaceae, capradaceae, malvaceae and some other families as we have seen earlier. Then in the crucifery, that is also called as the anisocytic or unequal cell. This type of stomata is found in the members of crucifery and several genera of Solanaceae. Then rubiaceae, in rubiaceae or parasitic or parallel cell, as we have seen earlier, they have a 2 parallel cells. This type of stomata is featured in the family Rubiaceae and its allied families. Then caryophyllaceous or diacitic, that is a cross cell. It is common in the members of Caryophyllaceae, Lamiaceae and Acanthaceae. And then the graminaceous type, this type of stomata are commonly found in the members of Poaceae and Cyperaceae. As we have already seen these type of stomata in our previous slides. So there are around 31 known types of arrangement of subsidiary cells in the stomatal complex of vascular plants. So there are around 31 types of arrangement of subsidiary cells in the stomata complex of vascular plants. At higher taxonomic level, these distinct pattern plays a role in the classification of several taxa. However, in some plant, the various types of stomata are present together. So in some, as we have seen earlier, there are the some common type, which is the one of the characteristic of particular families, as we have seen. Then the next is the trichomes. Trichome is also part of the epidermal cells and they are the outgrowth of the epidermal cells. So trichomes constitute the appendages of epidermal cells which may be non-glandular or glandular, and the non-glandular may be in the form of a simple unicellular cells or maybe in the case of a multicellular hairs that common in the Vesicaceae, Lauraceae and Moraceae, and in the form of a vesicles or peltate hair, as you can see in case of a olea or maybe flattened scales. So as you can see, this one is the simple unicellular hair that be showing the multicellular hair, and this one is showing the scale that present in case of a many, and these are the D that showing the calendula trichome of verbascum. Then E, that's showing the vesicular hairs of the atripex. Then peltate hair, that's F is showing the peltate one. Then stellate hair in case of a styrex, that showing in case of a this particular image. Then secretory glands of thymus, as you can see in this particular H figure. Then the stringing hair of utrica, as you can see in this particular image. So these hairs may be unicellular or multicellular, branched, may be glandular, non-glandular. So plant branch hair may be dendrite, as we have, and stellate, as we seen in case of our styrex, as you can say in this particular image. Then glandular trunculum may be sessile or stalk and present a variety of forms. As we can see, they may be have different types of variety. So as you can see, there are some special type of a trichome present in some genera, that is the maybe work as a identification tool in this particular genres. So you can see these are the number of types of trichomes. Yes, you can see this one is the unicellular. Then these all are the multicellular types with different form. This is the uni, then bobbed, coltrate, falcate, maniliform, then cylindrical. So these are the further many types, like this one is the stellate, radiate. So then in the glandular, they may be single celled or multi-celled, they may be T-shaped. So there's a number of type of trichomes, as you can see here, they may be dendroid or dendrotic in this particular image. So there are different types of trichome that may be work as an identification tool in some many genus or species. So as we have seen earlier that the trichome hold considerable promise in the systematics of angiosperm. As we have seen, the trichomes have been considerable help in the crucifery and especially in the genera Arabis and Arabidopsis. And the trichome characters are useful in the large genus, like in case of Astragalus, that has a more than 2000 species, and in the family Combretaceae, the trichomes are of immense significance in the classification of genera, species or even varieties according to stage. And the trichome are also diagnostic character of many species of Vernonia. So as we can see, it is very helpful in the identification of particular species. Now moving towards the stem anatomy. As Stage has shown that the anatomy of the stem can be used to distinguish the majority of its British species, like Dioscura species are distinguished on the basis of their stem anatomy. Then we can identify the Casuarina on the basis of their transformation of cortex into the translucent tissue. Then structures of stem of endodermis in the family such as the Atraceae and Pipraceae. Then the presence of bicollateral vascular bundle, that is arrangement in two alternate rings in case of the Cucurbitaceae, which is one of the diagnostic character of the family Cucurbitaceae. Then occurrence of cortical and medullary bundle in some families such as the Amaranthaceae, Chenopodiaceae and Nyctogenaceae are some of the features of taxonomic importance. And basically we identify the two groups of angiosperm, monocot and dicot, on the basis of their anatomy. As dicot have a ring arrangement of vascular bundles, as we can see here from this particular diagram, and in the monocot there will be no arrangement of such type. They have a scattered type of vascular bundles and they have a generally closed type of vascular bundle. They don't have a cambium and they don't show secondary coat, while the dicots have a open vascular bundles that have a xylem and they are arranged in a ring. So this is the one of the basis of classifying the two group, monocot and dicot, and we can easily identify these two groups on the basis of their stem anatomy. Now moving towards the next important characteristic, that is the nodal anatomy. Nodal anatomy has a considerable significance in angiosperm systematics, like it is also help in the identifying the primitive and advanced group, like the number of vascular traces entering the leaf base and associated types like lacunae left in the vascular cylinder of stem at the each node are distinctive for several groups, and the node may have a single gap. As you can see, they are termed as the unilacunar from a single leaf trace, or maybe they have a 3 leaf traces with the 2 additional common entering, or they have a 3 gaps, as you can see in this, that termed as a trilacunar or associated with the 3 leaf traces. So as you can see, these are the nodal anatomy with this one as A showing the unilacunar node with 1 leaf trace. In this B, you can see this is the trilacunar node with 3 lap traces, and the C, you can see this is one of the unilacunar node with 3 leaf traces. So, as you can see, the unilacunar nodes are found in case of laurels, caryophyllales, ericaceae, ebenales, primales, myrtles, and few tubiflory and astridae members of the angiosperms. Then, and multilacunar nodes are found in case of magnolias, piperales, crocodendrons, umbellales, and asterales. So, if we see the different types of nodes in different plants, as you can see, this one is the unilocular, which present in case of Asparaceae. Then trilocular present in case of Salix. This trilocular is present in case of Brassica. Then unilocular in case of Veronica. Then multilocular in case of Drumex. Then two-trace unilocular in case of Cleodendron. So you can see there is a quite variation in the type of a nodal anatomy, and according to Dixon, the correlation of nodal anatomy with some other features might help significantly in tracing the functioning of particular angiosperm. And the Philipson attempted to use the nodal anatomy as an aid of taxonomy. Then Sennett considered the trilocular node as a primitive and unilocular node and multilocular node as a advanced type. They said that all the unilocular and multilocular are originated from the trilocular nodes, and according to Paliwal and Anand, the majority of dicolor generally possess the trilocular nodes. They supported the Sennett view because as trilocular are primitive one and majority of the dicots have a trilocular. Then the genus Illicum has been separated from the Vintraceae because of the unilocular nodes and continuous pseudonephrosyl and the absence of granular material in the instrumental depressions. Then the next in the support of anatomy is the petiole anatomy. Metcalf and Chalk and Howard, according to the Howard, families, genera, even species may be identified by petiole characters such as their position on the stem, the presence or absence of stipules, its vascularization, nodal structure, and number of traces. There are some important features that help to identify the particular species. So, petiole anatomy of 64 species of Bhapi of Leguminosae and some species of Phlomis and Aeromostachys and Labiati provide clear support for the use of petiole anatomy in taxonomy for these genera. As we can see, these are the different types of, this one is the simple leaf that you can see, this is the Polypodium persculum, this one is the compound leaf, as you can see the leaflet or pinna, these are the one dioid leaf which is called as the unipinnately compound leaf. Then there is a different types of venation in the leaves you can see. So these are the different types of leaves. Then as you can see in this particular image, then the leaves, these are the xylems, then the vascular arrangement of the particular leaves. So if we see the leaves, then they maybe have a forked vein as in case of Polystrichum, then simple arrows in case of a Dahlia erecta, then recurrent vein in case of Theloptrica, or arrows with included veinlet in case of Cytomium. So you can see there is a variety. And then in case of a palmate leaf, this is a further dissected leaves. Then palmate leaf have the petiole and then divided in a lobed lamina. If we see the leaf anatomy, Koyama and Govindrajula studied the leaf anatomy of several species of Cyperaceae and formulated keys to identify the various species of Cypress and Purana. And leaf anatomy has been widely used in several taxonomical different groups such as the Euphorbiaceae, Cyperaceae, and Gramineae of angiosperms and Coniferae of gymnosperms. And the patterns of distribution of Sclerenchyma in Carex and Festuca have been used in the distinguish of the species. Then Sclerenchyma is also used in differentiating two genera of Velocea, that is the Velocea and Barbicillarium. So as you can see, the leaf anatomy is very important. This is the general figure showing the diagram of the leaf anatomy. This one is the dicot leaf and this one is the monocot leaf. And as you can see, these are the upper palisade tissue, upper epidermis, lower epidermis, palisade parenchyma, spongy parenchyma. As you can see, this is the epidermal layer. And here you can see the collenchyma, these are the epidermal cells showing the stomata, this one. So you can see here, this is the parasitic type of stomata, this is the guard cell has a parallel, subsidiary cells are parallel to the guard cells. As you can see, they have a number of air spaces. These are some general diagram of the leaf anatomy. And the next is the sclerates. Sclerates are the cells with very thick lignified walls. They are extremely rare in monocot, except in some genera of Araceae, Agavaceae, Ericaceae and few other families. And in dicots, they are more common in woody form than in herbaceous one. So Rao and Das have shown that the taxonomic value in about 30 species of Limonium. And then the next is the specialized cells and cells content. Like macroscopic characters of cell content such as starch grain in case of a Solanum tuberosum, protein bodies in case of a Cactaceae, then some cells have some plants have a specialized cells and they have some specialized cell content, like macroscopic characters of the cell content such as the starch grain in case of a Solanum tuberosum, protein bodies in case of a Cactaceae, albuminoids in case of a Leporti, then the large silica body in case of a Musaceae, Ericaceae and Zingiberaceae, then calcium oxalate crystal in case of Iconia and Allium, cystoliths in case of Amoraceae and Eutricaceae, teniniferous cells in case of Xyderaceae, important diagnostic tools and at time proof extremely helpful in delineating the species, genera and families. And the next is the wood anatomy. Would represent the secondary xylem constituting then the next is the evidences is the wood anatomy. Would represent the secondary xylem constituting the bulk of trees and shrub formed through the activity of vascular cambium. It is a primary consists of a tracheids and vessels. Tracheids are the narrow elements with tapering ends, imperforate at the ends and transfer of water and minerals occurring through the pit pairs. And the vessels on the other hand are the composed of vessel elements much broader than the tracheids and with perforation plates at the end of the cells. So vessel elements are joined end to end in the form of a long tube with the vessels. Then the perforation plate may be simple with single opening or may be compound with the several openings. The latter with the elongated opening in a roll-like ladder is known as the scalariform, or which is common in primitive in angiosperms. And if we see the wood anatomy, several features can be used to identify taxonomic group like vessel elements, vascular rays, axial parenchyma, presence or absence of storied wood, and the presence or absence of latex vessels, resins, crumb crystals. There are some features which can be used from the wood anatomy to identify a particular species. If we see a wood, it has a outer layer that is the bark, then they have a inner bark, it is termed as the bast, then the cambium which is actively divided cells, then you can see these are the medullary rays, then the outer wood is termed as the sapwood which is inactive and the hardwood which is active in nature, and you can see these are the number of growth ring according to the different types of seasons that separated out. So if we see the wood anatomy, and as all we have seen that the vessels are absent in case of a gymnosperms but present in the angiosperm. So we can identify the woods of gymnosperms on the basis of just our vessels. It is a commonly believed that there has been a progressive evolution in the angiosperm from tracheids to long, narrow vessel elements with slanted scalariform perforated plates to short, broad vessel elements with simple perforation plates. So those plants which have a number of multiple perforation plates, they are termed as the primitive, while the simple perforation plates are termed as the most advanced type. And if we see the wood anatomy has been contributed largely in the arriving at the conclusion that the Amentiferae constitute a relative by advanced group and that natales are not ancestral to the angiosperms. Studies on wood anatomy have contributed largely in arriving at the conclusion that the Amentiferae constitute a relatively advanced group and that natales are not ancestral to the angiosperm. Then Bailey concluded that the vessel in angiosperm arose from the trachea with scalariform pitting, whereas in natals they arose from the tracheids with circular pitting, thus suggesting an independent origin of vessel in these two groups of plants. And demonstration of vessel-less angiosperm, like in case of a Ventraceae, Trochodendroceae, also having other primitive features, have led to the conclusion that the angiosperm insects were vessel-less. Then the separation of Paeonia into distinct family Paeonaceae and Austrabelia into a family Austrobeliaceae has been supported by the studies of wood anatomy. And wood anatomy has also promoted the allocation of Amborella, Tetracentron and Trochodendron to their respective independent families. Then the placement of, that is the Myriceticala close to Lauraceae, and the delineation of different subgenera of Cricus of Fagaceae and non-inclusion of Calicanthaceae in roseales or metails are supported by the wood anatomy. Then the last in this anatomy section is the floral anatomy. Floral anatomy of Anonaceae, Calicanthaceae and Menispermaceae confirm that all these families are from the Ranunculaceae. Then uniformity in the floral vasculature of Solanaceae and Scophoriales suggests that both should be included in one single order, that is the Scophoriales. The separation of Paeonia from Ranunculus and inclusion under a separate family Paeonaceae was supported by the floral anatomical studies. Then formerly Cyperaceae and Gramineae were treated together in one order, but Hutchinson treated with separately in Cyperaceae and Gramineae because of their floral anatomy. Then Lilia, earlier included under the family Schuckelziaceae, was later separated into the independent family Liliaceae. So as we can see, floral anatomy further contributed to the taxonomy and they are work as evidences in this particular topic. So if we see the internal structure of flora, the floral has a different part like accessory organs like calyx, corolla, and they have a part of the perianth and there's a reproductive organ that are the main part of the plant, that is the stamens and gynoecium. These are the internal structure of flora. If we cut the anatomy of this particular plant, they are showing the resemblance with the other part of the flower, and by studying this we have concluded that the floral anatomy is very important for the different identification of particular species in the plant. So that's all for today in case of evidences of morphology and anatomy in case of a taxonomic evidences. Next we will study other evidences like cytological or embryological evidences in the next. So that's all for today. Thank you all of you for joining this session.